In terms of disclosures, this slide lists the various scientific advisory boards and grants that I receive. I should say that none of these represents any funding for any of the work I will describe today. Now, everyone in this room is very familiar with the metabolic syndrome. I like to view the metabolic syndrome as a cluster of disorders which are centered around insulin resistance. Of course, we're all familiar with the most common of these disorders, glucose intolerance and type 2 diabetes, as well as obesity. But of course, many other components of the metabolic syndrome are also linked to insulin resistance, including hypertension, dyslipidemias with accelerated atherosclerosis, hepatic steatosis, reproductive dysfunction, particularly in women with polycystic ovarian disease, increased risks of a number of cancer and Alzheimer's disease, as well as impaired longevity. And all of these disorders, including insulin resistance itself, are really created by combinations of genetic and environmental factors. Now, understanding these genetic and environmental factors is really central to eventually understanding the causes of these disorders and ultimately the best way to treat them. Some human studies have given us great insight to these genetic and environmental factors, and I just want to quote two very classic studies, which I think will help put this in perspective for this group. The first is a study done now 25 years ago by Claude Bouchard when he was in Canada, in which he wanted to ask the question, how much is weight gain genetically controlled? And to do this, he found 14 pairs of identical twins, and he put them in a clinical research center for 100 days, and he got them to voluntarily overeat 1,000 kilocalories a day, 6 out of 7 days of the week. And what he plotted in this New England Journal paper article was the weight gain in twin A on the vertical axis versus the weight gain in twin B on the horizontal axis. And what he showed was that there was a very strong correlation between the weight gain in one twin versus the weight gain in the second twin, and therefore the strong influence of genetics on weight gain following a high-fat diet challenge. But when I look at this slide, what I see is something also very important, and that is if you look at the twins on the left, they only gained 6 kilograms during this 100-day study, whereas the twins on the right gained almost 14 kilograms. That is, there was a twofold difference in weight gain among these twins, even though they were overfed exactly the same amount of food. Now, the second relationship is one that you also recognize well, and this is an example from the studies of Jerry Reven, who has looked for many years at the relationship between BMI and insulin resistance, in this case measured by something called steady-state plasma glucose, or SSPG. And what is shown here for 300 non-diabetic individuals is the strong positive correlation between BMI and insulin resistance. On the other hand, when I look at this, again, I see something that's very important to keep in mind, and that is that at any BMI, there are individuals who are very insulin sensitive. Take these individuals here at a BMI of 30, who are just as insulin sensitive as the leanest individuals, and other individuals who are very insulin resistant. So that given the same challenge, different individuals will gain different amounts of weight, and once individuals gain weight, their response in terms of insulin resistance will also show a very broad biological range, although positively correlated. Now, we got interested in this problem by looking at some of the mouse models we were creating to study the mechanisms of insulin action and insulin resistance. And many of these were created using mice of different genetic background. For example, the C57 Black 6 mouse and the 129 mouse shown here. And these mice, like the humans I showed you on the first slide, have very different propensities to weight gain and insulin resistance. For example, here you see the weight gain in a C57 black 6 mouse versus a 129 mouse, given the same 60% high-fat diet. And you can see that over the 18 weeks of this study, the black 6 mouse gains about 80% above its starting body weight, whereas the 129 mouse gains only 25% above its starting weight. This is despite the fact that the 129 mouse eats as much as the B6 mouse, and if anything, the 129 mouse is less active than the B6 mouse. So the B6 mouse is everybody's nightmare. I don't eat very much. I'm very active, but I still gain weight. And the 129 mouse is everybody's dream. I can eat all I want. I can lie around the cage all day, and I remain nice and lean. And in addition to their differences in weight gain on this high-fat diet, these mice also have differences in propensity to develop diabetes, whether when challenged with a high-fat diet or when challenged, in this case, with a genetic form of insulin resistance due to a heterozygous knockout of the insulin receptor and the insulin receptor substrate 1, IRS1. So if we look at these double heterozygous mice over their first 6 months of life, you can see that almost 90% of the B6 mice develop diabetes, whereas less than 10% of the 129 mice develop diabetes. Now, we were interested in trying to understand this because we thought that this would give us more insight into the nature of the differences in humans between weight gain and insulin resistance. And so we did first a genetic analysis of these mice. And we found that there were at least 4 genes on 3 different chromosomes that contributed to this difference. One of these was the gene for an enzyme called protein kinase C delta, a novel protein kinase C, which has an alteration in its promoter sequence, probably, that allows for different levels of gene expression of this gene. And here you see the levels of PKC delta expression in the liver of the B6 mouse in red and the 129 mouse in blue, first on Chow diet, where the B6 mouse has about a 50% higher level than the 129. And you can see when challenged with a high-fat diet, the B6 mouse has a further increase, whereas the 129 mouse has no change, resulting in about a 2.5-fold difference in gene expression for PKC delta. And what made this gene of particular interest to us was 2 things. The first is that when we looked in livers of humans who were obese or obese type 2 diabetics, we saw this same 50 to 100% increase in expression of PKC delta in the livers of these obese and diabetic humans. And the second thing that made this of interest to us is we knew that when we looked at the insulin signaling cascade with the insulin receptor, the IRS proteins, PI3 kinase, AKT, and this cascade of signaling events that lead to all the metabolic actions of insulin, some of which you heard described earlier in this meeting by Steve O'Reilly and some by Lou Cantley, we knew that PKC delta was one of the several novel isoforms of PKC which could serine phosphorylate the IRS proteins and inhibit its tyrosine phosphorylation and insulin action, particularly when levels of free fatty acids were high and intracellular diacylglyceroids were high. And this is illustrated for you here in an experimental model in which we've taken normal mice and we've either transfected into the liver PKC delta, the 4 lanes on the right, or a control adenovirus expressing green fluorescent protein, and we're looking at the level of serine phosphorylation of IRS1, and you can see that normally this serine phosphorylation is actually insulin stimulated, part of the way that insulin turns off its own signaling. But you can see that when you have higher levels of PKC delta, just 3-fold higher in this case, you get higher basal levels of serine phosphorylation of the IRS proteins, both in the basal and insulin-stimulated state. And because you have higher serine phosphorylation of the insulin receptor, you get decreased tyrosine phosphorylation of the insulin receptor. And this tyrosine phosphorylation is the critical phosphorylation for transmitting the insulin signal to downstream molecules like AKT. So as a result of this, you have decreased insulin-stimulated AKT phosphorylation. So changing the level of PKC delta by just 2 or 3-fold can actually create a moderate level of insulin resistance. And so at least one of the genetic factors that controls the difference between these two mice is PKC delta, with B6 mice having higher levels than 129. But there are other differences. For example, B6 mice also have higher levels of inflammation in their adipose tissue, which leads to insulin resistance compared to 129 mice. And at the same time, B6 mice actually have lower levels of brown fat or beige fat, energy-burning fat, which makes them more susceptible to obesity. So with these 3 genetic differences among these 2 strains, we can see some of the contributory factors leading to differences between insulin resistance and obesity in these 2 inbred mouse strains. Now, everything was going along just fine. We had our model with the B6 mice shown in red gaining more weight than the 129 in green, whether they be on Chow diet, the 2 lower lines, or on high-fat diet, the 2 upper lines, until a new fellow joined the laboratory. And this new fellow said, you know, I've repeated that experiment that the previous fellow did some years ago, and I get a different result. When I do this experiment, it is true that the 129 mice gain less weight than the B6 mouse on Chow diet, but when I put them on a high-fat diet, they gain every bit as much weight as the B6 mice. So now we had a conundrum. Is this the difference between the work of the 2 fellows, or is there something else intrinsically different in the way the experiment was done? And it turns out that there was something intrinsically different in the way the experiment was done. Because in the experiment shown on your left, both the B6 mice and the 129 mice came from Jackson Laboratories. And in the experiment shown on your right, the B6 mice come from Jackson Laboratories, but the 129 mice come from Taconic Farms, another major vendor of animals. And when we looked at this in more detail, it turns out that these 2 129 mice were quite different. So here, for example, is hepatomegaly in response to the high-fat diet, reflecting hepatosteatosis. You can see the B6 mouse on high-fat diet gets a lot of hepatomegaly. The 129 from Jackson, abbreviated 129J, gets practically no hepatomegaly. And the 129 from Taconic gets a kind of intermediate phenotype between the B6 and the 129 from Jackson. And when we look at glucose tolerance on the high-fat diet, the B6 mouse has by far the worst glucose tolerance, with fasting hyperglycemia and severely impaired glucose tolerance. But the 2 129 mice have relatively better glucose tolerance. In fact, the 129 mouse from Taconic, which actually gains more weight than the 129 from Jackson, has the best glucose tolerance of all. So we could say that in a way, the 129 mouse from Taconic is kind of an example of what we sometimes refer to as the fat-fit phenotype, an obese individual who seems to be relatively metabolically healthy. And this difference between these 2 mice was not at the level of PKC delta. PKC delta in both 129 mice was at a lower level than it was in the B6 mice. And in fact, insulin resistance was at a much lower level in both 129 mice, despite the fact that one becomes obese and one doesn't. So here's the HOMA-IR measurement of insulin resistance, which is very high in the B6 mouse on a high-fat diet and very low on the 129 from Jackson and the 129 from Taconic. So what is going on here? Is this genetics, epigenetics, or environment? Well, it turns out that these two 129 mice are a little bit genetically different. Initially, 40 years ago, they were exactly identical. In fact, Taconic bought its 129 mice from Jackson. But over 40 years of breeding, there have been some spontaneous mutations in both mouse strains. And also, occasionally, even in the best of these animal facilities, a rogue mouse gets in and deposits a few genes into the germline that are different from the original genes. So we know that between these two 129 mice, they are 99.97% genetically identical, but 0.03% genetically different. But of course, they could have other differences in epigenetics. They could have environmental differences. We know that the two animal facilities use different diets. One is vegetarian, one is both meat and vegetarian. They have different living conditions. And of course, now they may also consider the possibility that because of these differences, they also have different gut microbiota. So what is the gut microbiome? What are the bacteria in the gut? And why are they important for our consideration of the environmental causes of insulin resistance and metabolic syndrome? Well, studies over the last decade or so have really emphasized the fact that, as you know, we have bacteria throughout our intestine. They're present from the mouth all the way through the colon. In fact, in humans, there are estimated to be about 90 trillion bacterial cells, representing almost a kilogram of mass, of our body mass. And these bacteria, of course, play important roles in normal physiology and pathophysiology. They help us digest relatively undigestible food. They help us with some absorption and metabolism. And they may also be a source of inflammation, as we know, for those of us who've ever had Clostridium difficile infection. In the colon and in the feces, there are two major phyla of bacteria that are present. One are the firmicutes, and the other are the Bacteroides. The firmicutes are generally gram-positive organisms. The Bacteroides are gram-negative anaerobic organisms. And you can see that they comprise the majority of the bacteria in the colon. And what's of importance about this is that it was shown now more than 10 years ago, I think initially by Jeff Gordon, but also by many others, that in both obesity in mice and obesity in humans, these gut microbiota are different. They're less phylogenetically diverse, and in general, obese animals, here's the OB, obese diabetic mouse, compared to its lean littermate, obese animals and obese humans have higher levels of firmicutes and lower levels of Bacteroides. So this led us to ask the question, could the difference between these two 129 strains, and perhaps the 129 and the B6, be related to these gut microbiota, which might influence both metabolism and insulin sensitivity? So what do these different strains look like? Well, first of all, the ratios of firmicutes and Bacteroides, these two major bacterial phyla that are present in the feces and in the colon, are really quite different. So when you look, and we measure bacteria now in the microbiome, not by culture, the way we used to do and many years ago, but we actually measure it by sequencing the RNA or the DNA in the feces using high sequence techniques, 16S sequence techniques. And you can see that in B6 mice, the ratio of firmicutes to Bacteroides is about 60-40, as I showed you in the previous slide in lean animals. But in 129 mice from the same animal vendor, from Jackson Laboratories, the ratio for 129 Jackson is about 70-30. And for the 129 mice that come from Taconic farms, the ratio is almost 90-10. So these three mice have different gut microbiota. In fact, if you look at the actual level of the individual different species or taxa of microbiota, which are called, when you sequence them, operational taxonomic units, or OTU. This slide shows you all of the taxa that are at least at 1.1% of the fecal microbiota. You can see that in B6 mice, there's 149 taxa. In the two 129s, 169 and 178 respectively. These represent the different species of bacteria, and of these, only 59 are in common to all three strains, about a third. The others are found sometimes in two of the strains, like these two that come from Jackson have 49 others in common. But some of these bacterial taxa are unique to each strain of mouse. In the case 32 for the B6, 44 for the 129 from Jackson, and 75 from the 129 from Taconic. So even though these two 129 mice are 99.97% genetically identical at their own DNA, they're very, very different in terms of their gut microbiota. So, does this make a difference? Is this what's accounting for some of the physiological differences, and if so, how? So, what we decided to do to test this was to take these mice from these two vendors, these three strains of mice from the Jackson lab and Taconic farms, and to bring them into the same environment and breed them there for 3 generations. So we brought them to Joslin, and we bred them for 3 generations, fed them the same diet. We didn't put them in the same cage, but they were in the same room. And after 3 generations, we had what we called Joslinized mice. They had been environmentally normalized to be in the same environment. And now we decided what we would do is repeat the metabolic phenotyping and look at the gut microbiota to see how much we changed the gut microbiome and how much did we change the phenotype. Now, I won't be able to show you all of the data, but I just want to show you a couple of bits of the data to show you how the microbiome responds to a changing environment. So, for example, here is one species of Bacteroides, a Barnsiella species. And this shows you that in the B6 mice from Jackson Laboratory, the day they arrive into our laboratory, there is this level of 0.4 relative abundance of this bacteria. It represents about 0.4% of all the bacteria present in the fecal sample. When you put this animal on a normal chow diet, within 1 week, the level drops by half, within 8 weeks by 80%, and by 22 weeks, it's dropped by almost 99% from its starting level. And if we would have put these mice on a high-fat diet, the rate of change of that microbe was even faster, also going down, but almost 90% in the first week, and then down further during the remaining 22 weeks of this diet study. The 129 mice from Jackson Laboratory, in this case, happen to have roughly the same level of this bacteria when they arrive, about 0.4. And you can see again on chow diet in the solid line and in high-fat diet in the dashed line, they rapidly also re-equilibrate, at least over a period of weeks to months, to having these new levels of this bacteria. By contrast, the 129 mice from Taconic have almost none of this bacteria. They come with virtually no detectable level of this Barnsiella species, and this gradually rises while they're at Joslin to a level that's similar to, but not identical to, that in the other mice that I showed you. And this is just one pattern of change, but in fact you can see every pattern of change. You see bacteria that are going up, bacteria that are going down, some that go up on only high fat, some that only go up on chow, some that change in only 129 mice, or 129 mice from Taconic, and some that change in all 3 species. So that the microbiome is constantly remodeling in response to this new environment. And this heat map shows you the relative representation of these mice, first when they come from the original vendor, the 3 columns on the left, and then after they've been Joslin bred for 3 generations. And in this heat map, each horizontal line represents one bacterial species or taxa, and each vertical column, the strain of mice. And the color code, just to give you some idea of the changes, is on a log to the base 10 change. So dark black, or blue rather, dark blue means that there's essentially no detectable levels of this bacteria. And dark red means there's more than 1,000 copies per 10,000 bacteria present. That is, it's 10% of all of the bacteria in the gut. So you can see that when these animals come from the vendor, they have very few Bacteroides species, some though at very high abundance. These are represented here in these darker red colors, different in Jackson than Taconic mice. And then they have many different species of firmicutes and other bacteria, but that each of these mice has a different gut microbiome, just like I showed you in the Venn diagram. And after they've been at Joslin for 3 generations, that microbiome has completely remodeled. They now have many more Bacteroides species, different firmicutes. They have a more diverse microbiome. And yet, even after they've been in exactly the same environment for this 3 generations of breeding, they still don't have exactly the same microbiome, because what microbiota grow in their intestine is in part dependent on the host it's in, so host genetics controls the microbiome, just like it may control other responses that exist as well. So what happens as a result of this remodeling of the microbiome? Well, as I showed you in the earlier slides, when we look at the weight gain on Chow diet versus high-fat diet for these 3 strains of mice, the 129 from Jackson gains the most weight on Chow diet and the most weight on high-fat diet. The 129 from Jackson, the B6 from Jackson gains the most weight. The 129 from Jackson gains the least weight on both Chow and high-fat diet. And the 129 from Taconic is somewhere in between, being very close in weight gain to the B6 mouse. But after 3 generations of environmental normalization, these features change a bit. And in fact, now the B6 mouse, if anything, gains more weight when placed on a high-fat diet. And the 2 129 mice both are obesity resistant. The one that used to be obesity prone is now as obesity resistant as the other mouse. And not only have we made them more normalized with respect to their weight gain when challenged with high-fat diet, but we've made them more normalized with respect to many of their other metabolic features, including glucose tolerance. So here are glucose tolerance tests on Chow diet. On the left are the glucose tolerance tests in the B6 in red, the 129 from Jackson in blue, and the 129 from Taconic in green. You can see the very big difference in glucose tolerance with the 2 129 strains having different levels of glucose tolerance. But after they've been put in the same environment and have more similar microbiota, not the same, you can see that the B6 mouse still has the most abnormal glucose tolerance, but now both 129 strains have very similar glucose tolerance, indicating the important changes that this microbiome can make in glucose response. Now, I won't have time to talk about all the mechanisms, but we know that there are a number of mechanisms involved, that diet and genes get together to change the microbiome, and that this changes the barrier, the intestinal barrier, allowing endotoxins and cytokines to enter. It changes food and bile acid metabolism, resulting in changes in the circulating metabolome and metabolic signaling. That these come together to cause immune activation and metabolic regulation, contributing to metabolic syndrome in these animals. If we look at these different microbiota, we can find that in the gut, there are large categories of microbiota that are positively associated. The red color here means positively correlated with different metabolic phenotypes, all shown across the top of the slide. And the blue color means there are many others that are negatively correlated. So some microbes seem to be pushing us toward a metabolic syndrome, and others actually protecting us from the metabolic syndrome. And some of these correlations between microbiota and metabolic phenotypes are as strong as the correlations between any of our physiological hormone measurements and metabolic phenotypes. Here are 4 different bacteria that are highly correlated with either body weight, liver weight, blood glucose, or adipose inflammation. So how can we test whether these are really causal? Well, we can do two things that would allow us begin to understand which of these bacteria might be creating some insulin resistance or metabolic syndrome. One is we can try to modify the bacteria by giving treatments such as probiotics, or in this case, antibiotics. We can take these strains of mice and we can give them, for example, vancomycin, which kills most gram-positive bacteria, or metronidazole, which kills most anaerobic bacteria. And we can ask what happens to the bacteria in the gut. Here's the original bacteria, and here's the bacteria after vancomycin and metronidazole. So we've completely remodeled the microbiome again with antibiotics and differently in each of the 3 different strains. And we can ask, what does this do to metabolism? And you can see that when we give either vancomycin or metronidazole to B6 mice, we have an improvement in their random or fasted blood glucoses in this case, whereas the 129 mice, which have better blood glucoses, don't change. And we actually have improvement in insulin signaling. This is looking at the effect of insulin injection on insulin receptor and AKT phosphorylation in the liver. And you can see here's the insulin receptor line, here's the AKT line. At the bottom, I've simply quantitated for you the AKT phosphorylation. And you can see that here's the basal and insulin-stimulated response in control animals. In mice on vancomycin, there's an increase in basal receptor phosphorylation and AKT phosphorylation that you can see here. And on metronidazole, there's an increase in insulin-stimulated AKT phosphorylation. So we've modified insulin sensitivity. And we've done this, at least in the B6 mouse, by changing microbiota, and in this case, reducing the level of inflammation, both in adipose tissue and in the liver. And you can see the great decrease in TNF-alpha, IL-6, and F480, a macrophage marker, in the liver of these animals on both of these antibiotics. Finally, one of the one of the ways in which we can most directly prove this association is by actually transplanting the microbiome from one animal to another animal, in this case a germ-free mouse, and ask, can we fulfill Koch's postulates that these microbiota can actually produce metabolic syndrome? And we've begun to do these experiments, transplanting, for example, the microbiota from B6 and 129 mice into germ-free mice. And you can see that when we do this, the B6 mice bacteria microbiome causes these germ-free animals to gain more weight than the 129 mouse bacteria, causes them to have higher blood glucoses, and causes them to have more hepatosteatosis. So what I've tried to show you over the last 30 minutes or so is that obesity, diabetes, and insulin resistance are the result of interactions between genes and the environment. And that one component of the environment we now have to consider is this gut microbiome, which itself is the result of interaction between genes, diet, and other environmental factors, including where the animal lives, the provenance of the animal. Changing the microbiome by changing the environment, by changing the diet, by giving antibiotics, by fecal transplant, can certainly change insulin signaling and the pathophysiology of metabolic syndrome, and therefore understanding the interaction between insulin signaling and insulin resistance in the gut microbiome may provide us new ways to prevent and treat diabetes, obesity, and the metabolic syndrome. So I'm going to close now, and I just want to acknowledge the great fellows I've had in my lab over the years that have participated in this work, and in particular I've highlighted 4 names on this slide for you: Siegfried Usser, Timmy Herring, Olivier Bazzi, and Shihu Fujizaka, as well as the names of our collaborators who've done most of this work. Thank you so much for your attention, and thank you for this award.